So the allometric scaling, and I'm thinking specifically about clearance here, okay, how do you judge that actually that's the proper method? Like if you have a good allometric relationship?
There's some definite like enterohepatic recycling. That's a different species, have very different characteristic. So we can do with another species when you project it before it get to the human, based on your log correlation to see. For example, that CD40 show difference, but that one we recognize after human. Indeed, some cases they're not predicted well.
And for clearance, would you use just total clearance, or would you specifically use unbound clearance? You mentioned fraction of bound.
Depending on the protein binding.
Okay, so it's better to use unbound clearance.
Right, we would probably would test for each, for both. Yes. It's easier for lab exercise.
A couple questions, because we're doing some of this stuff with you and without you. So the hepatic cycling, enterohepatic cycling, you said there's differences between species, and that could be then hard. So which species do you find might be most predictive of whether or not such a thing might occur in humans? So that's one question. And second question, I have a ton of questions, so I won't hog the mic, but food effect. Do you see that more does that affect the enterohepatic cycling in some of the systems you're studying, whether or not the animal is fasted before you're doing an acute PK, or whether or not they are eating freely?
The first one, for the enterohepatic recycling, we recognize that mouse and gallbladder, rats do not. But still, probably also depend on the compound, depend on extent of that EHC. So nowadays, the doctor always says that nobody has the crystal ball to predict that. But for this one, yes, enterohepatic recycling is more complicated. The model is probably recently developed. Yeah. And for the second one, the food effect also depends the absorption. First, the physical chemical nature of the compound. If that triggers the bile secretion, that will be affected more. Okay. Right, fatty food or something. And secondly, also depend on absorption mechanism, whether the particular compound is very fast absorbed, or there's a slow one, may be different. So probably you have a better answer.
Well, I don't know specifically for the food effect on the enterohepatic circulation, but I think if you eat under the food, the bile probably will excrete more. I'm not sure, because then they're probably impacting the bile secretion when you're eating something. I don't recall any data, because when we're doing the food effect studies, we never look at the drugs with enterohepatic circulations. The food does have effect on the drug absorption, but how that impacts, I think the drug eventually is excreted from the bile and will be hydrolyzed basically in the small intestine. Actual how impact on that, I don't recall anything where we did anything with the enterohepatic recirculation, but there was of course, it has effect on the drug itself.
For your compound, do you have data with the fasting and with the food?
We have so much data.
Yeah, that probably interesting to analyze to see, test the hypothesis.
One point that the enterohepatic recycling is really difficult to predict, because it also depends on the microbiome of the body gut. It will metabolize the drug differently. Even between humans, they are very different. So that's what I learned from another presentation. So, but there is one software that could do prediction in DHC. It is the Jasper. They have a model. So I have one question for Dr. I'm with the human when we like cannot like under that condition?
The site of action is in the tongue, so we apply the patch on the tongue. How do we do it? We have to let them rat to sleep. We can't just have them to awake. So we anesthesize the rat. So they are sleeping, and actually they are sleeping. So we are putting these patch onto the tongue. And apparently they stayed around there. Okay. And the problem is that we applied for there for about 4 hours. In the hours, because this first time we checked the relation, no one had tried this before. So we actually applied there. And our collaborator, he said he wants to apply for 4 hours. So I was thinking 4 hours is too long, but we actually did it 4 hours and it was stayed there. We only have one accident that we can't find the patch at the end of the experiment, but most of those patches stayed onto the rat. And the particular rat, we can't find the patch, I guess it's swallowed, but it was sleeping. So it was we don't know what happened, but I guess it probably the rat swallowed on that. So we also be cautious about that application once we remove the patch. And apparently there is a bunch of drugs on top of the surface of this tongue. And if we're not careful, a few of the rats, when we did the initial study, they actually have the blood concentration shoots up. And we noticed that because these drugs are still on the tongue surface, and they basically swallowed. So we later on in the later control rats, we actually wash the tongue before we collect more samples. So that way it's everyone's same, because if they swallow after that, it means you have oral administration versus once applied to the patch. So we control that point on that.
Dr. Liang, I have a question for you. When I saw your slide, after 4 hours, you have 1.3% of dose in the tongue, and the remaining in the patch only fractional percentage. Where are those other doses?
Oh, that's a good question. Oh, yeah.
The tongue, you have a table showing 4 hours in the tongue.
Oh, yeah,
1.3% dose.
Yeah, that's what I was talking about. The
fraction.
remaining, because what we done is that after 4 hours, we remove the patch. And then we collect the tongue, and we wash the tongue out of it. And we analyze the drug in the tongue itself. And that's only 1.3%. And we also analyze the ones residual we collect back. Okay. And then it's not coming to 100%. So we were looking at where the rest of the drug goes. One possibility, it comes into the systemic circulation, which is not much of it, because we look at the plasma level is not that high. So another possibility is that it's on the surface of the tongue, we wash it out of it. And we happen just wash, we were looking at the tongue itself, we did not collect that amount. And we was assuming that probably that's where the leftover.
Would that be possible in a different part of the cavity?
I don't think so, because we don't know. We actually tried the tongue application, sublingual application, and the buccal application. And we tried preliminary rats, and if we find out that the application to the sublingual, it gets a fastest absorption. But since it's for the tongue, that's why we only focus on the tongue application.
It urges me to say a few things about drug development. There's lots of drugs or drug candidates that are in development, but the current estimate is that for every 100 projects that start, there's only one kind of drug. And also, there are accounts out there that for every 10 animal studies that are deemed to be successful in preclinical studies, only 9 of them fail in clinical trials, somewhere in clinical trials. So there is a very high attrition rate in drug development. So where does PK/PD, or what are the major weaknesses in your opinion, on the PK/PD aspect that have been a challenge in translating preclinical findings towards clinical studies? What could be done to improve predictability of animal studies to launch successful drugs?
We're looking at basically some ideal compound. So out of these optimized compounds, we usually will, once we do an IV study, oral study, we have some idea, an in vitro study idea with this drug eventually will go or not going. But in case of some lead compound that this is very potent, I have to do something, then we dig deep. we're looking for different delivery systems. We're looking more into, even though the drugs has low bioavailability, metabolized extensively. If we can characterize them, we can still push on that point now. But from our point of view, of course, we're looking for ideal compounds, water soluble, but most of that may not be. But it doesn't mean that we have to kill the compound, though. We're looking at different aspects. If the compound metabolizes erratically, we don't know the mechanism of it, we can figure it out. Or the drug has, if it's unpredictable, mainly if it's unpredictable, or it's nonlinear kinetics-wise, or it's not soluble, those are main issues we are encountering there. And then it's all these facts, I believe, is all combined in making the decision whether we're going to push this ahead or not. Yeah.
This is definitely a very important question for industry. Now they are saying if you need to fail that candidate, try to fail it upfront, early. So that's how the preclinical IND enabling study is very important and have to be very critically evaluated. And there's several things. One is biodistribution to potential toxicity. That's the FDA concerned the most. And secondly is for in vivo, you see the exposure and efficacy. That one may not be linear, translate cross species. And also whether that's effective delivery, the drug. Now it's monitoring for the drug in the circulation. Most of the cases they correlate with those level site of action, but not necessarily always true. So that's a few, when you review, if you're in industry, that's why they encourage you to try to be critical to see those points with your preclinical data.
Question on one of the models you presented, the rat perfusage model.
Yeah.
Yeah, and basically at what time of the characterization of a new potential drug do you use that model, and how do you use the data? And also, like, it seems very experimentally involved. Like, so is it a very low throughput assay to do? Thank you.
We actually mainly we're looking at how the drug permeable in terms of if the drug intended for the oral administration, for example. So two things we want to look at: where the drug metabolizes, or how the drug passes through the small intestine. So our animal study, of course, can find out about whether it's high or low, but we can't tell whether the drug is permeable or not. So we use, well, authentically, we have the cockatoo cell permeability study. We can do that. And we also have the ones we presented here is the in situ perfusion model. So we actually make the rat to sleep and clean all the GI tract, and we close both sides of it, infuse the buffer solution containing the drug, passing through it, and then in the meantime we collect from the beginning to the end what's the concentration change about it. If the drug concentration changes, we assume that the drug being passed through the small intestine. So that is, from our point of view, is pretty feasible and provides some more information, because in the meantime we collect the biosamples, which is coming through the liver metabolism. We also collect blood samples, and it's pretty informative study, and it's not very difficult because it's pretty straightforward. And we set up the whole thing. We have animal protocols lined up to go, so we don't have to wait a long time for approval process or something like that. And it's very informative, because out of that we can know pretty much what's going on there, plus with the drug metabolism. So it's a fairly useful tool, actually. Yeah.
Also, the KICO2 will enable us to identify whether there's any transport involved in uptake or to the efflux.
Right. Yeah.
Hi. Would you mind talking a little bit more about the center itself, how it operates, whether I should consider the modeling first and then the experiments, those kinds of things, and also fee, how much you charge versus collaboration, the practical things.
Sure. So the center, how operates, we operate very similar to the other co-facilities sponsored by CIPRITs. The operation usually, if an investigator has an interest in using our facilities, you contact us, and we will initially set up some kind of conference call or in-person meeting to find out what's going on about the particular compound. So different stages. So first, if I come out of it, I have this compound highly potent out of the cellular molecular level, and I want to move ahead. So that's at the very beginning. Or if you have done everything already, I just want you to analyze some sample. I have some clinical sample, or I have some clinical, some study, preclinical study we done already with a mouse model, but I have some blood samples for you to analyze it. We do that one. Or even more that I have, you can establish something, and I have some data, and just go ahead and analyze some PK/PD simulation to figure out what's going on with the kinetics versus the other one. So depending on different stages, so we will usually sit down, we'll look at what stage usually kicks in on that. So usually we need some kind of actual meeting to see what area we will kick in. So in other words, the earlier one or the later ones. Most of our, right now, our investigators are mainly in the early stage, that they have a series of compounds, I want to do something on that. So that we usually ask them to give us a little bit time. So usually we ask them to give us information of the drug. So if you want to go for animal study, we can go earlier. So it takes these are time consuming, and give us some compounds we can establish an assay method. Because once these are done, when the actual study comes in, we can kick in right away. Or we need some formulation study. We do have a big area for the formulation. We characterize it. We have a pion system. We actually can do quite a lot of physical chemical checkings of a new compound, pKa's, log P's, pH stability profiles, in quite a few of the in vitro characterizations on their compounds. So the different areas we involved in that. So once we have this settled, we have a fee structure, but it depends. Because in other words, some they want, we have quite a few of these collaborators coming to us that I have the grant, I want to resubmit a grant, incorporate something. Can you give us a letter of support? We are very happy to write all these letter of support for the investigator to apply for the grants. And once the grant comes in, they can use our core facilities. Some of the investigators have their own funds. We have some collaborators coming from different institutions. They just want us to enter a sample, they pay us, and that's it. And some are more toward what we want to do more in the study with you guys, and we can publish together. And then in the meantime, they will use facilities. So it depends on different cases. We have we have different ways of operating in this area. Yeah.
So the case that I'm thinking about on a daily basis right now is essentially trying to pick the lead compounds to get more data around, and we have rat PK data. And I kind of, I've been reading up on allometric scaling and how variable it is and that type of thing. And I kind of wanted to know, to reach the desired once a day oral dosing, how far can extended release formulations take you? And I know this is a hard question with nothing other than rat in hand, but I'm looking and I'm there's got to be a floor of half-life or something like that under which you go, okay, don't look at those, toss them out. Assuming the multitude of compounds are potent in vitro and in cell-based assays, when do you not try to engineer your way around something to get to that great holy grail of once a day dosing? Is it just you can't answer that question with that information that you currently have? It's possible. Yeah.
Yeah. So your question basically that when do we start looking at this sustained release dosage formulations? Yeah. Yeah. So.
How much?
Right.
Yeah.
Wow.
You give up and move on to the next compound?
Well, yeah, that's a good question. But I think the first focus is that for a compound itself, do we know enough about this compound? Okay, and do we have a possibility of this compound to move to the IND? If never ever moved out of IND, why do we bother for all this kind of problem? Of course, if the drug has very rapid half-life, 1 hour, 2 hours, 30 minutes, you do have to think about the sustained release formulation of it. But usually, I believe not in that early stage. I don't know, Dr. Chow, how do you think? That early stage, we still don't know what the drug itself, and it's just too early to kick into that. But usually when we are pretty much sure the drug is safe and effective to move to the IND, then we have to think about it. Do we have the patient dose 3 times, 4 times a day, or do we need to develop something more of reasonable? So drug companies usually kick in at a fairly late stage than that early stage that we are looking at now at this point.
To add a point to that, industry actually move faster to eliminate some drug candidates, but nowadays they have a whole shelf of those good candidates didn't develop further. So that's not easy question to answer. So that's how the term they said repurposing, repositioning. A lot of those potentially good candidates didn't get developed further. So on the other hand, you mentioned
and it's not uncommon for the drug company to develop immediate release formulation first to the market, and then along the way will develop different type of formulation, control release, sustained release, any kind of stuff, so they can patent them, even increase their patent life too as well. So that's evolving. Once the drug is approved, they can do some bioequivalency studies or some other easier ones to go on. It's very often.